These are the receptor tyrosine kinase proteins. These are the tyrosines. These are the signaling molecules. These are the inactive relay proteins, and these are the phosphates that will attach to the tyrosine later. Today we're going to discuss the signal reception in tyrosine kinase.
This process occurs on the plasmic membrane.
A kinase refers to an enzyme that catalyzes the transfer of a phosphate group.
Here the receptor protein functions as a tyrosine kinase that catalyzes the phosphate group from ATP to the amino acid tyrosine on the substrate protein.
This process is highly complex due to there being more than 10 pathways for this one pathway to occur.
Before the signaling molecules bind, the signal receptors exist as single polypeptides.
Then the ligand connects to the two polypeptides, creating a dimerization.
The dimerization activates the tyrosine kinase region of each polypeptide, adding a phosphate from the ATP, making an ADP.
The P's add on the sides of the tyrosine, making it fully active and a phosphorylated dimer.
Now the inactive relay proteins connect to the phosphorylated dimer.
Lastly, two cellular responses are transferred out, and the activated tyrosine will activate ligand proteins.
When tyrosine kinase does not fully function properly, it often causes problems with cell division. The phosphorylation of kinases in a cell is very important for transduction regulation. It helps to communicate signals within a cell and regulate activity such as cell division. They also help the activation process of signal transduction from a phosphate group to adenosine triphosphate.
Fibroblasts are a type of cell that synthesize the extracellular matrix and collagen and is involved in wound healing, which uses a tyrosine activity in its cellular matrix. The tyrosine kinase activity plays its part in cellular transformation. It has been demonstrated that phosphorylation of a middle T antigen on tyrosine is associated with cell transformation, a change that is similar to cellular growth or reproduction.
Tyrosine kinase are particularly important today because of their implications in the treatment of cancer. A mutation that causes certain tyrosine kinases to be active has been associated with several cancers. The pathways have genetically altered to favor the mutating cancer cells. Imatinib is a drug that is able to bind to the catalytic cleft of tyrosine kinases, inhibiting its activity.